Welcome to Simplified UPSC by PPP, where we listen, learn, and grow together. Detailed lecture notes in English and Hindi, plus practice questions for prelims and mains, only on our Telegram channel. Link in description. Like and subscribe for more.
Welcome back to the Deep Dive. This is the place where we don't just list facts. We really try to build knowledge.
Exactly.
We know you're tackling one of the most demanding exams out there, and rote memory just isn't going to cut it. You need the connections. Those links between the core science, your NCERT curriculum, and the kind of high-yield integrated facts that actually show up on papers like the UPSC.
And that's our mission today, really. It's laser-focused. We're going to cut through the noise and just distill the most critical concepts.
Using the Class 11 biology text as our blueprint.
Right. We're zeroing in on excretion, biomolecules, metabolic pathways, and then immediately linking those fundamentals to the facts you need for, rapid recall and application.
Deep understanding, high-yield results.
That's the goal.
Okay, so before we jump in, and it's quite a journey today, we're going from the body's microscopic filters to the fundamental forces of the universe. We do need to talk about support.
Yeah, this is really important.
If the analysis we provide here, if it's helping you streamline your prep, which is just incredibly demanding, please consider joining the paid membership of the Simplified UPSC by PPP YouTube channel.
That support is just... It's
Wow.
critical. It's what allows us to dedicate the time to create this kind of curated content for you.
It really is. And to all of our existing members, a huge heartfelt shout out.
Thank you so much. Your support is genuinely the cornerstone of this whole operation. It fuels all the research, the synthesis, everything.
And for everyone listening, please do remember to hit that like button and share your comments. We read them, and we use that feedback to shape every single deep dive.
We really do.
Okay, let's unpack this. Our central theme for today is the metabolic cost of selectivity. And we're starting where selectivity is, well, arguably most intense.
In the kidney.
In the kidney, the body's ultimate filtration system. We're diving into NCERT Chapter 19, focusing on the plumbing of the nephron, starting with the proximal convoluted tubule, the PCT.
Right. And, people often focus on the glomerulus, the initial filter.
Sure, that's where it all starts.
But that's just a passive strainer, really. The PCT, on the other hand, that's the engine of reabsorption. Structurally, it's defined by its simple, cuboidal, brush border epithelium.
The brush border, that's the key phrase, isn't it?
What?
What does that structure let it do so well?
It creates a massive surface area. Think of it like tiny folds, microvilli, just everywhere.
Purely for efficiency.
Exactly. And it needs that surface area because its job is immense. The PCT actively reabsorbs nearly all the essential nutrients that were just filtered out.
We're talking virtually 100% of things like glucose and amino acids.
100%, plus the vast majority of key ions like sodium, Na plus up.
So that's the reclaim function. It sounds like a triage center deciding what's absolutely precious and must be retrieved immediately.
That's a perfect analogy. And remember that retrieval is often active transport. It requires a significant amount of ATP.
Our first connection to the theme, the metabolic cost.
Precisely. But the PCT's job isn't just reclaiming. It's also about balance. It actively secretes ions like H plus and K plus back into the filtrate.
Why does it do that?
To maintain the blood's very delicate ionic and acid-base balance. It's a two-way traffic system, all to maintain homeostasis.
Okay, so after the PCT does its high-energy retrieval and balancing act, the filtrate moves on. And this is where the mammalian kidney gets its, legendary status.
Its ability to conserve water.
And produce that highly concentrated urine. It's a masterstroke of adaptation for life on land.
Absolutely. And this whole efficiency is enabled by something called the countercurrent mechanism.
Okay, break that down for us.
It involves the flow of the filtrate in the loop of Henle and the flow of blood in the vasa recta. Those are the blood vessels running right alongside the loop.
And the key is the counterpart.
The defining feature is the counterflow. The fluid going down one limb is moving in the opposite direction to the fluid in the adjacent limb.
Why is that opposing flow so important? Why not have them flow in the same direction?
Well, if they were parallel, the concentration gradient would equalize very quickly.
Oh, okay.
But the counterflow makes sure that equilibrium is never reached. Instead, it allows the kidney to build and maintain a crucial and pretty extreme osmolar gradient in the surrounding tissue.
And this gradient isn't a minor difference, is it? It's huge.
It's massive, and this is the high-yield number to remember. The gradient ranges from about 300 milliosmoles per liter.
Osmo 1.
Right. near the outer cortex, and it quadruples, rising all the way to 1,200 mosmol 1 deep in the inner medulla.
So that steep gradient, that's the environmental leverage that the rest of the system uses.
It is the driving force. That hypertonic environment is what allows the final segments, the DCT and the collecting duct, to pull the maximum amount of water out by osmosis.
Passively.
Passively. The collecting duct dips deep into that super salty hypertonic medulla, and it uses that 1,200 osmol 1 gradient to its full advantage. The body ends up concentrating the filtrate about 4 times over. It's a beautiful, efficient water recycling plant.
That makes perfect sense. A complex structure is an investment that pays off big time in survival. Now, let's bridge that. We see this incredible complexity in the kidney, and it's all driven by tiny molecular pumps. So, speaking of the molecules that run the show, let's zoom out to proteins, the universal building blocks.
Right. Proteins are the workhorses. They're structures, transporters, enzymes, everything. And if you want to understand a protein, you have to start with its primary structure.
Which is what exactly?
It's simply the sequence, the positional information of the amino acids linked together by peptide bonds. Get that sequence wrong, and the entire function can just collapse.
What's fascinating is the huge prevalence of some of these proteins. We have 2 key abundance facts that come up a lot.
They do, because they're so significant evolutionarily. The most abundant protein in the animal world is collagen.
Structure, bones, skin, tissue.
Exactly. But if you zoom out to the entire biosphere, every living thing on the planet, the most abundant protein is ribulose bisphosphate carboxylase oxygenase.
Thankfully abbreviated to Rubisco.
Thank goodness for that.
Rubisco, the main enzyme in photosynthesis. It literally feeds almost all life on Earth. So it makes sense it would be the most abundant. But there's a huge irony here, isn't there?
A profound evolutionary irony. Rubisco, even though it's the most abundant enzyme, is also incredibly inefficient.
Because of the oxygenase part of its name?
That's it. It often confuses oxygen for carbon dioxide, and when it does that, it kicks off a process called photorespiration, which just wastes energy. The planet's most dominant enzyme is, well, it's pretty clumsy.
So plants have to make massive amounts of it just to get the job done.
Exactly. It's like solving a problem by just throwing enormous resources at an imperfect tool. But most enzymes, unlike Rubisco, are masters of efficiency.
So how do they get that catalytic power?
They work by lowering the energy barrier, the activation energy, that's needed for a substrate, S, to become a product, P.
And they do this by binding to it.
Yes, they temporarily mine the substrate in their active site, forming a short-lived enzyme substrate, or ES complex. They don't add energy, they just find an energetic shortcut.
A faster path for the reaction.
Millions of times faster than it would be on its own. It's metabolic acceleration.
Okay, let's shift gears now into the plant world. Let's talk about how their growth is managed by plant growth regulators, or PGRs.
Right, and they fall broadly into 2 camps. Promoters like auxins, gibberellins, and cytokinins.
And inhibitors.
And inhibitors like abscisic acid.
The discovery stories here are actually pretty cool.
They're fascinating bits of science history. Take auxin. Its function was first noticed by Charles and Francis Darwin.
The Darwins.
The Darwins. They saw that the tip of a canary grass coleoptile bent towards light, phototropism, but it was F.W. Went, decades later, who actually isolated the chemical responsible from oat seedlings.
A step-by-step process of observation leading to isolation.
Exactly. And gibberellins have an even more dramatic story.
The foolish seedling disease.
The Botany disease in rice, yeah. Reported by E. Kurosawa way back in 1926, he found that a filtrate from a fungus, Gibberella fujikuroi, caused rice seedlings to grow ridiculously tall and spindly and then just collapse.
And the chemical was gibberellic acid.
That was it. A great example of how studying a plant disease led to a major discovery.
And on the inhibitor side, we have the famous stress hormone.
Abscisic acid, ABA. It's known for inducing dormancy in seeds and promoting abscission, the shedding of leaves or fruit. It's basically the plant's emergency brake.
We can't forget ethylene either, this simple gaseous one.
Ah, ethylene, the agent of ripeness.
Right.
It's made mostly by aging tissues and ripening fruits. It promotes ripening by boosting the respiration rate, a process called the climacteric rise.
So that's why you put a banana in a paper bag to ripen it faster.
Yeah.
You're trapping the ethylene.
You're trapping the ethylene it's already producing.
Okay, connecting plant life back to global cycles, let's return to our theme, metabolic cost. Let's talk nitrogen metabolism.
A process with a truly staggering energy investment.
And it's so critical.
It's one of the most critical cycles on Earth. Nitrogen is essential for proteins, for DNA, but the N2 in the atmosphere is it's unusable by most life.
Only prokaryotes can do it.
Only specific bacteria and cyanobacteria are capable of N2 fixation.
And the high-yield fact here is the cost of that conversion, isn't it?
It absolutely is. In symbiotic fixation, like what rhizobium bacteria do in the root nodules of legumes, the energy input is tremendous.
Okay, what are we talking?
You need to spend 8 ATP molecules for each single ammonia NH3 molecule produced.
Wow. 8 ATP for 1 NH3. That really reinforces the theme. If a task is selective and essential, it comes with a high metabolic price tag.
It really makes you respect the sheer energy the biological world invests just to get usable nitrogen. And the enzyme that does it, nitrogenase, has a major weakness.
An Achilles heel.
It does. Nitrogenase is extremely sensitive to oxygen. The reaction needs strict anaerobic oxygen-free conditions to work.
But wait, the bacteria living inside a plant root nodule, which has a vascular system, there has to be oxygen there.
There is. So the question is, how do you manage this? How do you perform this delicate chemical reaction in an environment that should kill your key enzyme?
So what's the solution?
The host plant and the bacteria collaborate. The host nodule produces a special oxygen scavenger. It's called leg hemoglobin.
Like hemoglobin, like the hemoglobin in our blood.
Chemically very similar. Its job is to mop up any stray oxygen inside that nodule, keeping the nitrogenase enzyme safe and functional. It's a brilliant biological solution to a fundamental chemical problem.
Fantastic. Linking that cellular detail right back to the global ecosystem. Okay, let's hit the final stage of our deep dive. We're moving out of core biology and into some of those classic PYQ-style integrated facts.
All right, let's start with economic botany and ecology. Bio-fertilizers.
Right. It's critical to know which organisms are which.
It is. The sources highlight the cyanobacterium nostoc and the algae Spirogera. Nostoc is especially important because it can do both photosynthesis and N2 fixation.
Perfect for places like rice paddies.
Exactly. Waterlogged fields. It adds a huge amount of fertility. And it's vital to know that something like agaricus, a mushroom, is a decomposer, not a biofertilizer in this context.
Good distinction. Okay, shifting to geography, a classic linkage question. What is the one critical prerequisite for the world's best fishing grounds?
The prerequisite is the meeting point of warm and cold oceanic currents.
And it's not just about the temperature mixing.
No, it's about density. When these currents meet, the differences in temperature and density cause deep, nutrient-rich water to rise to the surface. It's a phenomenon called upwelling.
And the upwelling is the key to the fish.
That's it. The bottom of the ocean is full of nutrients from decayed matter. Upwelling brings it all back to the sunlight, feeding the phytoplankton.
Which are the base of the entire marine food web.
Exactly. No upwelling, no massive fishery.
And on land, what defines the environment of an equatorial forest?
The key characteristic is the presence of tall, closely set trees that form a continuous, dense canopy.
And that canopy is everything.
It's so thick it severely limits the light that reaches the forest floor. The whole structure of the forest, its biodiversity, humidity, temperature, it's all defined by that continuous cover overhead.
Okay, let's jump to general science. Optics and physics. 3 atmospheric phenomena that are fundamentally optical illusions.
Okay, the 3 categorized as optical illusions are the apparent larger size of the sun at dusk, the color shift of the sun at dawn, and the twinkle of stars in the sky.
Wait, I get the sun looking bigger, but why is the color shift an illusion? And the twinkling stars?
They're illusions because they're caused by atmospheric refraction and scattering, not by a change in the light source itself.
I see.
When the sun is on the horizon, its light travels through a much thicker slice of atmosphere. This bends the light and scatters the blue wavelengths away, leaving only the reds and oranges for us to see.
Making it look redder and sort of flattened.
Exactly, an illusion of its true color and shape. And stars twinkle because their point source of light is constantly being bent by moving layers of atmospheric density. It's the atmosphere distorting the image, not the star flickering.
That context is what turns a fact into real insight. Okay, speaking of light, what 3 phenomena come together to make a rainbow?
That requires 3 specific actions, all happening inside a raindrop. You need refraction, as light enters the drop.
Okay.
Then internal reflection, as it bounces off the back of the drop.
Right.
And finally, dispersion, which is the splitting of the white light into its component colors.
All 3 working together. Got it. Lastly, let's lock down 2 key facts about the 4 fundamental forces.
First, electromagnetism. This force is behind all chemistry, light, magnets. And the key thing is, it acts only on particles that have an electric charge. No charge, no electromagnetic interaction.
And the second one.
The weak nuclear force. This is the fundamental interaction responsible for processes like beta decay. In other words, it's the force that causes radioactivity.
What a sweep. We've gone from the high energy cost of the PCT to the molecular dynamics of Rubisco, the nitrogen cycle, and right into the integrated facts on ocean currents and the physics of light.
And the theme holds up all the way through: the metabolic cost of selectivity.
It really does.
Every precise essential function in biology, from reclaiming sodium in the kidney to the 8 ATP needed to fix one nitrogen atom, is driven by a huge energy investment. Life operates by making these selective, high-cost decisions.
It makes you think about ATP as this universal currency, driving every single selective function across the entire biosphere.
It's the cost of doing business.
Whether that business is maintaining a gradient in the kidney or building, a biological snorkel like late hemoglobin in a root nodule.
Something to ponder as you continue your studies.
Absolutely. Thank you for joining us for this deep dive. And if this kind of synthesis is helping you make those critical connections, we do ask humbly that you consider supporting our work.
Please subscribe and think about joining the paid membership of the Simplified UPSC by PTP YouTube channel. That support is what allows us to keep bringing this level of analysis directly to you.
And a huge final thank you to all our wonderful existing members.
You are the ones who make this possible every single week.
Please also remember to like the video and share your valuable comments below. Keep learning, keep connecting the dots.
And we'll see you on the next deep dive.
Thanks for watching. Follow us on Telegram and Instagram. Don't forget to like, share, and subscribe.